大家好. So, but anyway, 5 minutes is like kind of short, but I'll try to finish and make you understand what we are doing as much as I can. My name is Dr. Kumar Surupa. I am head of business development for Kongwin Biopharm. We are a new biotech company that is researching and developing tumor ablation techniques, which are quite effective for various kinds of solid palpable tumors. Most of us, we know that tumors are kind of managed with surgical approaches, chemotherapeutic approaches, radiotherapy approaches. Nowadays, ablation devices, targeted therapies, and precision medicine is kind of developing in the market to control tumors. So we are an ablation technique. We are researching on an ablating way. And let me show you how we are different from any of these therapeutic approaches in the market. Our drug, our proprietary molecule, paratoluene sulfonamide, is a small molecule that has been independently developed by our company. So it is an injectable, as you can see. And this is the first liquid ablating drug that has ever received approval for treating airway tumors in non-small cell lung carcinomas. Last year, we received our approval for mainland China. Now, in lung cancers, what you can see over here is how the tumors are formed inside the airway lumen. So in the first case, it is quite easy for the current approaches to easily ablate the tumor. But dealing tumors that are extraluminal or of mixed types is still a big challenge. Through our approach, we could easily locate the tumor with existing diagnostic devices, and we can easily reach out to the tumor and inject our drug. Now, as you can see, this is a case from our phase 3 clinical trials. In the first picture, you see that the airway lumen is totally occluded by a tumor. So restoring airflow is the priority. Now, using a bronchoscope, we reach out to the target site and we directly inject our drug inside the tumor. Necrotic effect starts within the same day or maybe in a couple of days. So before you administer the second round of injection, you are supposed to remove the necrotic tissue and then again inject the drug. So the idea over here is to inject and remove the necrotic tissue until the whole area is clear of any tumor tissues. In the second case, the necrotic tissue has been removed before administering the fourth dose, right? So here, there is no defined procedure on when to inject and when to remove. It depends on how the drug is reacting to the tumor and how the physician is able to inject the drug. It requires a certain technique. So, from the clinical studies that we have so far, we can say that our drug is safe. It is not damaging the luminal walls, as you can see. It is quite effective in initiating the necrotic effect, and at the same time, it is having a broad applicability. In our phase 2 studies, we have found out that our drug is quite effective on oral tumors, head and neck tumors, liver cancers, different sorts of palpable tumors. So this is how our pipeline looks. We have, as said, we already received the approval in China, but in the same time, we are conducting phase 2 clinical studies in Taiwan. We have an orphan drug designation for treating tracheal adenocytic carcinoma with the US FDA, and we are initiating studies to manage malignant pleural effusions. This indication has already been licensed out to an Australian-based distributor. And another interesting thing in our company is the veterinary application, which is a new segment. So after receiving approvals from the local regulatory bodies, we have also kind of started evaluating the efficacy of our drug on treating tumors in canines.